Hello and welcome back to another edition of WIP's Biochemistry. This time we'll be discussing the cholesterol biosynthesis. Now, cholesterol is classified under sterols, under lipids. We can have a quick recap of the structure as well as functions of cholesterol. So this is the structure. It's a heterocyclic structure, 3 hexacycles, 1 pentacycle, and this is often referred to as cyclopentanosteroid ring system. And there is an OH group also, the alcoholic group, so that's why cholesterol, and a double bond also is there. Now, let's see the functions. One of the major function is as component of membranes. As being a component of the cell membrane, it adds to the fluidity of the membrane, as well as it fits to the gaps of phospholipids, so that it can restrict the entry of hydrophilic substances across the membrane or to the cytoplasm. Now, another function, a major function, is as being precursor of some of the important biomolecules. One of them is bile acid and bile salt. And in fact, conversion of cholesterol to bile salt is one of the modes of excretion of extra cholesterol in our body. And bile salt is having its own functions. And another function is as precursor of vitamin D. Vitamin D, there is 7-dehydrocholesterol, which is a common precursor of vitamin D as well as cholesterol. But cholesterol can be converted to 7-dehydrocholesterol also. The reference is given though before it is converted to vitamin D. Another is as a precursor of steroid hormones like cortisol, corticosteroids. So, as well as sex hormones, cholesterol is a precursor to steroid hormones also. Now, the sources of hepatic cholesterol. Normal serum value of cholesterol is 140 to 200 milligram per deciliter, and maintenance of this level is important. Both hyper and hypocholesterolemia is not good for your health. And how this is maintained? No, dietary cholesterol. A normal typical daily dietary intake of cholesterol is about 200 to 300 milligrams. And this dietary intake means, in fact, dietary cholesterol is transported to liver hepatocytes by chylomicron remnants, which are formed from chylomicrons, the lipoproteins. Now, further, cholesterol will reach hepatocytes from extrahepatic tissues by reverse cholesterol transport also, that we will discuss in a future lecture. And apart from this, or the main source of hepatic cholesterol is de novo synthesis. A normal adult typically synthesizes about 1 gram, so that makes 1000 milligrams of cholesterol per day. And how? So that is the point of our discussion in this class. Now, almost all cells in the body are capable of synthesizing cholesterol, but the primary site remains liver, and the site of synthesis is cytosol. Even though smooth endoplasmic reticulum is also involved. The secondary sites are given here: adrenal cortex, ovaries, testes, mammary glands. Obviously, these are the tissues where cholesterol is put into use. For example, adrenal cortex, ovaries, testes, where steroid hormones are produced. Mammary glands, where fat deposition, cholesterol deposition is there. And what is the overall equation? So this is it. The carbon source for cholesterol biosynthesis comes from acetyl coenzyme A. The energy comes from ATP. It's also breaking the bonds of acetyl coenzyme A itself. That process itself drives some of the reactions forward. And cholesterol biosynthesis involves some reduction steps also, and the electrons for reduction is donated or comes from NADPH. So these are the raw materials of cholesterol biosynthesis. Now, all the carbons in cholesterol are formed from acetyl coenzyme A. Now, acetyl coenzyme A, most of the acetyl coenzyme A are formed inside mitochondrial matrix. Now, obviously, the result of fatty acid beta oxidation is acetyl coenzyme A. Then the result of carbohydrate metabolism, we know that glycolysis gives rise to pyruvic acid, which enters mitochondrial matrix and gets converted to acetyl coenzyme A. And usually, acetyl coenzyme A combines with oxaloacetic acid and oxaloacetate to give rise to citrate, which goes through TCA cycle. Or if acetyl coenzyme A gets accumulated, can go through this process also, so as to give rise to ketone bodies. With the increased fatty acid breakdown, this process can also take place. But when citrate gets accumulated, and the TCA cycle do not need further citrate. Citrate can be used for the synthesis of both fatty acids as well as cholesterol. So citrate has to go out of mitochondrial matrix, and this is done with the help of mitochondrial citrate transport protein. So citrate goes out of mitochondrial matrix and reaches cytoplasm. Yeah, like this. Where it gets converted to acetyl CoA and oxaloacetic acid with the help of the enzyme ATP citrate-lyase. Now, acetyl CoA is out of mitochondrial matrix, it reaches cytoplasm. Now, acetyl CoA can be used for synthesis of fatty acid here. So, fatty acid synthesis also involves the same process as well as cholesterol synthesis, the same thing. So, it is obvious that if a high carbohydrate food is taken, that can be channeled for the synthesis of lipids. So, that is what happens here. Now, acetyl CoA formed, and we can see the nutshell of cholesterol biosynthesis here. What happens next is acetyl CoA gets converted to acetyl CoA by condensation of two acetyl CoA molecules with the help of acetyl CoA synthase. Then HMG-CoA is formed with the help of HMG-CoA synthase, and further HMG-CoA reductase. It's an enzyme which is present in smooth endoplasmic reticulum. You'll see the detail in the further slides. The HMG-CoA reductase that actually reduces HMG-CoA to give rise to mevalonate, and further steps give rise to cholesterol. So this is actually the nutshell of cholesterol biosynthesis. We'll see it in detail in the further slides. And the entire process of cholesterol biosynthesis can be seen as divided into 5 phases for convenience of study. The first phase is synthesis up to HMG-CoA, and the second phase is synthesis up to mevalonate. Let us see. So the first 2 phases are coupled here. So mevalonate, up to mevalonate is shown in this figure. Now, 2 acetyl CoA which got into cytoplasm from mitochondrial matrix, they give rise to acetyl CoA with the help of the enzyme acetyl CoA synthase. And further, one more molecule of acetyl CoA come into play to give rise to HMG-CoA, which is 3-hydroxy 3-methyl glutaryl coenzyme A. And water molecule is taken, and the enzyme involved is HMG-CoA synthase. The extra coenzyme A portion is split off, and the next step is reduction of HMG-CoA, which is carried out by HMG-CoA reductase. This is the most critical or rate limiting enzyme, or the step in the entire process of the committed step in the cholesterol biosynthesis. And you can see here that it's a reduction process. It involves 2 molecules of NADPH which donates electrons. And further, here this is also shown here that bile acids, cholesterol, mevalonate all are having negative effect or inhibitory effect, as well as the medication statin, which is used to treat hypercholesterolemia or patients having high cholesterol. So statin actually acts as a competitive inhibitor of HMG-CoA reductase, whereby reducing the synthesis of cholesterol. So this is also shown here. The structural aspects can be studied here. Acetyl CoA is split off, further acetyl CoA, and then HMG-CoA, and finally mevalonate. So what we have with us now is mevalonate, and we move to the third phase. And the third phase is formation of isoprenoid units. So up to the formation of isoprenoid unit is the third phase. Mevalonate is a 6-carbon compound, 6 carbon, and all these 6 carbons have come from acetyl CoA units. That is, 3 acetyl CoA units are involved actually in the synthesis of mevalonate. So that means 6 carbons, and this 6-carbon mevalonate undergoes a series of phosphorylation steps which involves kinase, ATP splitting. So this is the phase. This is the third phase, which finally gives rise to isopentenyl pyrophosphate, which is a 5-carbon compound. And this 5-carbon compound, because the final step here, that's shown here, is a decarboxylation step. It's a decarboxylation step which removes a carbon from this particular compound, 3-phosphate. Yeah, 3-phospho, 5-pyrophosphomalonate. Now, you can see here that this compound is a 5-phosphomalonate, then comes 5-pyrophosphomalonate, which means 2-phosphate back-to-back arrangement, then one more phosphate on the third carbon and this pyrophosphate on the fifth carbon. That means 5-pyrophosphomalonate. Now, when actually what happens here is that when more and more phosphate groups are added to the molecule, this molecule becomes more and more negative and so hydrophilic, and the molecule remains soluble in cytoplasmic fluid. So that is also achieved here so that's why phosphorylation is done, or that is the effect of phosphorylation. Now finally, we have got what we have got here is isopentenyl pyrophosphate. It is basically it's an isoprenoid structure. So this is the first isoprenoid structure that is formed during the synthesis of cholesterol, and from now on, so this also is having 5 carbons. Now on, head to tail condensation starts to occur in the next phase. The process up to the synthesis of squalene is included under the fourth phase. What happens is that actually squalene is having 30 carbons in it, and the isopentenyl pyrophosphate starts the process. The fourth phase is having 5 carbons in it, and condensation of 6 such isoprenoid units, which are actually formed from isopentenyl pyrophosphate itself, I can see, give rise to squalene. So 5 into 6, which means 30 carbon squalene is formed. And how? Let us see. See, this is isopentenyl pyrophosphate, 5 carbon. It actually, first of all, it isomerizes to give dimethyl allyl pyrophosphate, which is again an isoprenoid unit. And 2 molecules of dimethyl allyl pyrophosphate condenses. So this is head to tail condensation. It gives rise to geranyl pyrophosphate. It's a 10 carbon compound, obviously, and this takes up another 5 carbons, and then those 5 carbons are taken from isopentenyl pyrophosphate, another molecule of isopentenyl pyrophosphate. So that makes 15 carbon farnesyl pyrophosphate with the loss of a pyrophosphate. Now again, 2 molecules of farnesyl pyrophosphate, which means 15 plus 15 combines to give squalene, and it's a reduction process involving NADPH, electron being supplied by NADPH, and with loss of the last of the pyrophosphate molecule here. So with the removal of, what happens here is that with the removal of pyrophosphate from farnesyl pyrophosphate, the last of the pyrophosphate, the product squalene, actually it becomes hydrophobic and incapable of remaining dissolved in cytoplasm. But this is actually solved by the interference of carrier proteins which remain attached to squalene and further products. So what happens is that these molecules remain dissolved in cytoplasm, and the process can be carried out further. So now what we have is this squalene, a 30 carbon compound, and we know that actually cholesterol is having 27 carbons only. Now this fifth phase involves cyclization of squalene to give rise to the parent Lanosterol, which is a 30 carbon compound, and that parent steroid compound which gives rise to 27 carbon cholesterol. So this is the process. Squalene, a 30 carbon compound, first of all gets reduced with the help of NADPH to give squalene epoxide, or squalene 2,3 epoxide, and the enzyme involved is epoxidase, and further cyclase enzyme is involved, which gives rise to Lanosterol. And Lanosterol, it remains a 30 carbon compound which gives rise to cymosterol as 27 carbon, desmosterol at 27 carbon, and finally cholesterol. So this process is actually it takes place in 19 steps. So it's not a single simple step. It involves 19 steps. It's an elaborate process, and not much of it is clearly understood also. It involves a number of enzymes also, like as shown here. So this is what happens. Finally cholesterol is formed. Now it has to be noted here that squalene 2,3 epoxide is actually it is also the parent compound of stigmasterol, the steroid of the plant steroid, as well as the fungal steroid, namely ergosterol. Both of them are derived from squalene 2,3 epoxide by different pathways. And finally here, cholesterol. Now, let us summarize what happened. So these were the 5 phases which we discussed. You can just pause and go through it. Thank you.